Welcome to our presentation on how greens can alter genes, by myself Ali and my colleague Fazela. We hope the following slides will suffuse a sense of awareness into the modern development of medical treatment and scientific research. When we mention genes, let's consider the Brussels sprout, for example. It's small, unassuming, and ostensibly good for you. However, this is no mere side dish as you'd imagine. It is now thought that through the modernity and development of science, that greens can alter genes in ways which are completely foreign to us, and will ultimately alter our gene expression. Now, let's take a look at how standard gene expression happens at a molecular level.
In 1958, Francis Crick came up with the theory of the central dogma, a fundamental part of molecular biology which states that DNA is transcribed to RNA, which is translated to protein. There are three main events that make up the central dogma: replication, transcription, and translation. The central dogma describes gene expression from DNA to protein. The process of gene expression is used by all organisms as far as science knows. This includes eukaryotes, prokaryotes, and viruses to generate the macromolecular machinery for life. There are two main stages of gene expression: transcription and translation. However, transcription also includes RNA splicing, which we will also be discussing during this presentation. Transcription is the transfer of the genetic information from the archival copy of DNA to the short-lived messenger RNA. Transcription happens in the nucleus of the cell, if it has a nucleus. If the cell lacks a nucleus, then the mRNA is immediately translated without additional processing. Firstly, the hydrogen bonds in the DNA break and the DNA double helix unzips. Only one strand is used as the template to make an mRNA copy called an antisense strand. RNA polymerase enzyme binds to the antisense strand on the DNA. The initial binding site is called the promoter region. RNA polymerase then matches a base on the DNA to an RNA nucleotide by complementary base pair binding. The new nucleotide is added to the elongated mRNA strand. RNA polymerase moves along the DNA, separating the strands and assembling the mRNA. Hydrogen bonds then reform the double helix. When RNA polymerase reaches the stop codon, it stops making mRNA and the completed mRNA molecule is released from the RNA polymerase DNA complex and can begin translation. In eukaryotic cells, this means first moving from the nucleus into the cytoplasm. In prokaryotic cells, ribosomes can bind and begin translation before polymerase has completed of the new mRNA strand. The protein-coding genes of a eukaryote typically contain regions of DNA that serve no coding function. Non-coding regions, called introns, interrupt the coding regions, called exons. When the gene is transcribed into RNA, both the coding and non-coding regions are copied. However, a eukaryotic cell has a mechanism for removing the introns from RNA. In a process called RNA splicing, a newly transcribed RNA molecule is cut at the intron-extron boundaries. Its introns are discarded and its exons are joined together. RNA splicing occurs within the nucleus before the RNA migrates to the cytoplasm. In the cytoplasm, ribosome translates the RNA, which now contains the uninterrupted coding information into protein. Translation is a process that takes the information passed from DNA as messenger RNA and turns this into a series of amino acids bound together with peptide bonds. It really is a translation from one code, a nucleotide sequence, to another code, the amino acid sequence. RNA translation occurs on the ribosomes, which are found outside of the nucleus attached to the rough endoplasmic reticulum. Firstly, the ribosome binds to a very specific region of the mRNA called the promoter region. This is just in front of, or upstream from, the sequence of mRNA that will be translated into the polypeptide code. Transfer RNAs are bound to specific amino acids. The anticodon on the tRNA has to match the codon on the mRNA. Each transfer RNA that binds to mRNA at the ribosome brings in the next amino acid. A peptide bond is formed to the previous string of amino acids, elongating the polypeptide chain. The ribosome moves to the next three-letter code on the mRNA and the process repeats. The ribosome continues until it hits a stop sequence. Then it releases the polypeptide in the mRNA. The polypeptide then forms into its native shape and starts acting as a functional protein in the cell.
Fragments of plant RNA have been found swimming in the bloodstream of humans and cows. Studies by Shenyu Zhang of Nanjing University shows that these plant RNAs muffle our gene expression. It was tested on mice and results showed a rise in cholesterol levels. This gives the notion that food can be developed into medicine. One molecule of particular interest is microRNA, often expressed as miRNA, which are tiny strands of RNA between 19 and 24 nucleotides long. It's a wise idea to question whether all our microRNA strands in our blood are actually synthesized by our cells or come from food instead. The team drew blood from 31 Chinese women, men, and cows. They treated the sample with sodium periodate, an oxidizing agent that modifies miRNA so that it cannot be sequenced. Crucially, it leaves the plant versions completely untouched. Zhang found that some 30 plant microRNAs were floating in the blood of people and cows. Emphasis was placed on miR168A and miR156A. These are abundant in rice, cabbage, and cauliflower. Since Chinese people have a high intake of rice, and considering that these microRNAs aren't destroyed whilst cooking, it can be concluded that those in human blood came from the food that they actually eat. From a series of experiments, Zhang found that these microRNAs are actually changing our gene expression. Put simply, since it causes a change in cholesterol levels in mice, so it must therefore cause changes beneficial for humans in treating abnormality. This does raise an important question, however. Do the gene firms insert in genetically modified crops change the health of people who eat them? This finally opens the possibility of designing diets and plants with therapeutic effects. As of now, Zhang is conducting research on whether the microRNAs of a specific Chinese herb are able to knock out the influenza virus. We hope that this presentation has brought some deep insight into what can be the future development and innovation of treatment.